Once upon a time, in a picturesque tropical bay, life was good. The water was clear and the food plentiful. The environment and humans didn't just survive, they thrived. However, life was so good that little by little, the human population grew, tripling in just over a century. So eventually, human pressures upset the delicate balance within the ecosystem. The problem is the excessive use of fertilizers to feed rapidly expanding crops, poor management of manures from livestock farms, along with aging or inadequate wastewater infrastructure to process the amount of waste produced by the growing population. This has inevitably resulted in large loads of nutrients like nitrogen and phosphorus reaching the waters of the bay. The result? Too much of a good thing becomes a bad thing. While nutrients are great for growing our crops, excessive amounts of nutrients enable algae in the water to grow out of control, like weeds in your vegetable garden. Scientists call this process eutrophication. What's the problem with algae, you might ask? Living algae are usually not a problem. The problem starts when the algae die. Bacteria that consume the decaying mass of algae remove the oxygen in the bay by respiring it, making the water suffocating for all other animals. The consequences to the bay are dramatic. Too much nutrient turned what was a productive and biologically diverse habitat of the many into the sick and degraded habitat of the surviving few. This is how you create a dead zone. What once was an idyllic bay with clear water has turned into a smelly soup of rotting algae. And as if this wasn't enough, people have started getting sick from eating seafood from the bay. Very sick. This happens because some types of algae can produce toxins that accumulate in fish and shellfish. These events are known as harmful algal blooms. This story is, unfortunately, a common one. The number of dead zones in our ocean environments has increased from fewer than 50 reported globally in the 1960s to well over 500 today, and it has come at a steep price. Dead zones don't damage just the environment. The community and the economy suffer too. Millions of dollars are lost every year due to lost fisheries, lost tourism, and additional healthcare costs. Much of the nutrient pollution comes from land-based sources, and countries have recognized that controlling this is a crucial part of fixing the problem. The first step to reducing nutrient pollution is to measure the amount of nutrients that we discharge in the environment and their source. However, we need to know something about how to measure the scale of the problem so that we may be able to influence our communities and industries to make needed changes. So, let us now explore how the science is helping us to do this. The most common means to relate the amount of nutrient input to the coastal environment and possible negative impacts has been by measuring chlorophyll A, the green pigment present in all plants and algae. Chlorophyll A can be measured from satellite or directly in the water. Hence, chlorophyll A acts like a kind of marker or indicator of total algal biomass and nutrient over-enrichment. So greater concentrations of chlorophyll A in the ocean means higher algal biomass, which is a symptom of high nutrient concentration. However, the use of chlorophyll A as an indicator has its limitations. An important one is that it cannot distinguish between algal blooms that grew thanks to natural nutrient inputs and those algal blooms that grew due to land-based nutrient pollution. For example, natural processes such as upwelling can bring nutrient-rich deep waters to the surface and trigger algal blooms, hence increasing the amount of chlorophyll A in the water. Measuring chlorophyll A tells us about nutrient concentration, but it doesn't tell us about its source, making chlorophyll A a poor risk indicator for such events. How can we overcome this limitation? Research tells us that the concentration of 3 key nutrients is essential in determining the likelihood of the growth of harmful algal blooms versus harmless algal blooms. Land-based nutrient pollution adds excessive amounts of nitrogen and phosphorus to the waters. Silica, another important element that also determines that balance between harmful forms of algae and harmless forms of phytoplankton, instead originates only from natural processes, such as the weathering of rocks. Here is where things get interesting. Diatoms are a group of phytoplankton that uses silica to produce its casing. A balance between nitrogen, phosphorus, and silica, like you would have in an upwelling area where nitrogen and phosphorus are relatively low compared to silica, indicates a balance of diatom and non-diatom phytoplankton species. This is the ideal condition you would find in a non-polluted environment. However, an excess of nitrogen or phosphorus relative to silica results in the dominance of non-diatoms, including flagellate algae. Flagellates are often undesired because they tend to be associated with coastal eutrophication and include toxic species. So, by measuring or estimating the concentrations of nitrogen and phosphorus relative to silica, we can determine the contribution of land-based nutrient pollution on eutrophication and harmful algal blooms. We can do this using the Index of Coastal Eutrophication Potential, or Icep. The Icep can be calculated by comparing the concentration of nitrogen, phosphorus, and silica with the nutritional requirements of phytoplankton. It estimates the amount of phytoplankton growth in excess of what could be attributed to diatoms and must, therefore, occur in non-diatom phytoplankton species that don't require silica to grow. A negative Icep value indicates a healthy ecosystem dominated by diatoms. Positive values indicate conditions where the land-based pollution may be degrading the marine environment and favour non-diatom algae. Large positive values indicate areas where non-diatom algae can thrive and significantly degrade the environment. The Icep, therefore, signals whether diatom or non-diatom phytoplankton species are likely to be favoured and how much non-diatom growth you can expect. It is important to note that coastal eutrophication can occur even when Icep values are negative. Icep is just an indicator of how likely non-diatom phytoplankton blooms are to occur. Icep is calculated by computer models that estimate nitrogen, phosphorus, and silica loading. These models describe how Icep is affected by land use, climate, and other factors. This information is useful for countries and agencies who wish to reduce damage associated with coastal nutrient over-enrichment. The Icep is used alongside existing indicators of coastal ecosystem health and water quality, including chlorophyll A and other measures that countries are already using. So why does all of this matter? Countries are obliged to report on the status of pollution of the marine environment under the Sustainable Development Goal 14. Under Target 14.1, the Icep is the official measure to be applied as indicator of nutrient pollution. The Icep will be combined with a marine plastic index to generate a more comprehensive measure of marine pollution. With the development of the Icep, countries will be better able to relate the quality of their coastal waters to nutrient pollution. With the support of IOC, UNESCO, and the United Nations Environment Programme, countries will be assisted to report on Icep by implementing the methodology at the local level or across transnational boundaries with shared watersheds and coastal waters, along with the implementation of measures to reduce the nutrient pollution of coastal waters. Our governments, industries, farmers, communities all need to work together to tackle the problem of nutrient pollution. Better understanding and better indicators of coastal conditions provide opportunities for better solutions to coastal environmental problems. This will help us move into action to achieve the Sustainable Development Goal we set, to prevent and significantly reduce marine pollution of all kinds, particularly from land-based activities, including marine debris and nutrient pollution. Follow the link to learn more about Icep.